to my cutest and bravest little champions. I hope everyone who is watching this video is doing absolutely good. And if at all someone is not doing that great in their life, please not worry. We all are there for you. Trust your instincts, stay calm, stay positive. Everything will fall in its place. Just keep on giving your best, and if there is something else that cannot be solved as of now, give it some time. Yeah? And I also know that some of you might be in this phase of preparatory exam. For some of you, it might not have started. So it's for all of you that we are giving a video of must-do topics in biology, my dear children. So today we have a special guest as well, which is very, very special for me. Our own biology NCERT. I hope everyone has seen it once in their lifetime at least. So yes. So today chapter wise I am going to tell you the must-do topics. Along with that, sometimes I might open this book with you also and tell you what's important from here by giving you the page number as well whenever I can. Okay. So let's get into it. The first chapter that we have carries 13 marks, my dear children, and that is Principles of Inheritance and Variation. So it should be the first chapter for your preparation as well. 13 marks is something that we need, right? Now the first thing, monohybrid and dihybrid cross and its ratio in Pisum sativa. So the ratio of monohybrid cross is given on page number 57. If you see over here, the phenotypic and genotypic ratio is given, my dear children. It's very, very, very important. You should be knowing, okay, 3:1 is the phenotypic ratio, 1:2:1 is the genotypic ratio, and things like that. And also, then after that, it is given about dihybrid cross. So dihybrid cross, 63 page number, if you can see over here, again the ratio is given, that is 9:3:3:1. So these ratios, how do you get it, and everything is important. They will ask you to show the monohybrid and dihybrid cross. Okay, so have a quick revision for that. Now 14 characters of Pisum sativa, again something that is very, very important. All the 14 characters are given up over here. If you see this particular thing, okay, here also you have, here also you have. I'll recommend you go with this, which is given on page number 54. Why? Because this is pictorial representation. It's in the format of what? Pictures. So you will be able to remember it in a much better way. Okay. Now incomplete dominance is the next thing. In incomplete dominance, what do we have an example? Snapdragon, my dear children. What do we have? Snapdragon. Again on page number 60, if you can see over here, there is snapdragon with pictorial representation. Red flower, you're crossing with white flowers, you get pink flowers. Then the pink flowers, when you're crossing, you're getting all type of flowers. Again, the ratio, the phenotypic and genotypic ratio, this is very unique in incomplete dominance. The phenotypic and genotypic ratio is the same, that is 1:2:1. Go through it. Again, very important. Okay, then we have multiple alleles example, that is the blood group. For multiple alleles you have different example, but I will restrict you to study about, give more attention to what the blood groups, that is A, B, O and things like that. The genotypic, the thing is given over here on page number 61, my dear children. Okay, page number 61, if you see, there is a box. Right. Now over here you will be finding what? All the genotypic, genotype for all the blood groups as well. So that is really, really important. Now rediscovery of Mendel's work. Rediscovery of Mendel's work is given on 65 page number, if you see over here. In this particular paragraph, that is in 1900, 3 scientists, De Vries, just above this particular picture, you can see this particular paragraph is really important. Go through it. Okay, there they have mentioned about De Vries, Correns, Schemark, all of them, they independently rediscovered Mendel's work. So this question comes in the form of MCQ. This is why I am telling you that. Okay, and then Walter Sutton and Theodore Boveri, notice that chromosomes and the genes, Mendel was saying, all of them, they have similarity, some similarity. So this question again can be asked, Walter Sutton and Theodore Boveri. It has been asked previously. So it's just for you to cement things up, I'm telling you that. Yeah, and now you're coming to linkage and recombination. So on page number 67, the first part where linkage and recombination is given, this whole particular paragraph is really, really important, my dear children. So they will ask you, for example, okay, if two genes are closely located, will they show higher recombination or lesser recombination? So if they are closely related, obviously they will be showing what? Less recombination. Recombination means new combinations could be formed. So if they are closely linked, if you and your best friends are too close to each other, you will not have any other best friend there. okay, you would not give that chance for any other friend to come in that zone. You get it? Similarly, linkage also happens like that. If two genes are very closely linked, there is less amount of recombination. So over here they are given the example, okay, if you go like 4 lines above it, you will be seeing 37.2% recombination, 1.3% recombination. I just want you to go through all this thing. This is very, very important. Okay, linkage and recombination. Now you talk about sex determination in insects, birds and human beings and honeybee. All together it's important. From page number 69 to 71, there is nothing that you can avoid. Okay, you have to read it really, really carefully. It's very fun thing. Okay, it's very, very fun thing. Haplodiploidy is seen in honeybee. Now that is something that is very much important. I will write it over here, that have come so many times. Okay, what is it, children? It is haplodiploidy, which is shown in honeybee. Okay, now male heterogamety, as in the case of human beings, males produce X and Y. So don't you think they are producing two different type of gametes? So this is known as male heterogamety. Okay, now there is female heterogamety, that is shown in the case of birds, Z and W. Okay, that means this would be a female. So they are producing two type of gametes, one can have Z, other can have W. You get it? So that are examples that you can see over here in sex determination. Those things are really, really important. Okay, so I hope that is also done. Now, okay, everything will blink. Yeah, okay. Now we have Mendelian and chromosomal disorders, my dear little children. So Mendelian and chromosomal disorders, if we talk about that. Okay, now you see this image on page number 72, where they have given boxes, circles and stuff like that. They will be giving you such boxes and asking you what does it indicate and things like that. So you should be knowing about that, a little about pedigree. So that thing I missed somehow. So you should be knowing about pedigree. Okay, and then what? If you're talking about Mendelian and chromosomal disorders, all of them are equally important. Okay, all of them are equally important, but in Mendelian I will recommend not to miss color blindness, and hemophilia. Very, very important diseases. Color blindness and hemophilia, you can get certain numerical, not exactly numerical, but yeah, numerical based questions on it as well. If you have seen my Brahmastra series, you might be seeing that there are so many questions around it. So you should be knowing about that. And now also in Mendelian you have thalassemia and sickle cell anemia. Thalassemia and sickle cell anemia. Sickle cell anemia is a qualitative disorder, my dear children. What is it? Okay, I might mess it up while writing. What is it? Qualitative. It messes up with the quality of hemoglobin. Thalassemia is a quantitative disorder. So that question is usually asked, which one is qualitative, which one is quantitative. In sickle cell anemia, what happens? So you should know that one particular base pair is being replaced. This is also an example of point mutation, my dear children. Okay, so diseases, none of them you should be missing, but please give more priority to the ones that I am showing you over here: color blindness, hemophilia, okay, thalassemia and sickle cell anemia. Both of them are blood related diseases, but they are different. You should be knowing whether which one is sex related, which one is autosomal. So both things that you see over here, these are X-linked diseases. Okay, sex linked diseases. So you should be knowing about that as well. Talking about chromosomal disorders. Okay, so here I have mentioned thalassemia and sickle cell anemia. Difference, why? Because qualitative and quantitative one that I was saying. Okay, same thing that I mentioned over here. Now what? I think one slide is missing somehow. Okay, anyways, so I'll put down over here if it is missing. That is why I took the book also, because I don't want to miss you guys to miss any of the single information. Now you might have different numbers also, page numbers also, which is why I won't be using the NCERT now, but just telling you. So my dear children, over here in chromosomal disorders, there is something like aneuploidy. You should be knowing about this definition of aneuploidy and also the diseases Turner syndrome, Klinefelter syndrome and Down syndrome. Okay, Turner syndrome, Klinefelter syndrome and Down syndrome. These are the 3 chromosomal disorders given in your NCERT textbook. So you should be knowing about that. You should be knowing about their karyotype as well. What is there? For example, in the case of Klinefelter syndrome, they have one extra X chromosome. So it will be 44 autosomes plus XXY. You get this? This is the karyotype. So you should be knowing the karyotype of Klinefelter's as well as Turner syndrome as well as Down syndrome. Okay, so these are about the diseases. Aneuploidy, something very important, please not miss. Now you have something else also, which is pleiotropy and polygyny. Pleiotropy's example is phenylketonuria. Phenylketonuria. So you should be knowing about that. Polygyny, its example is skin color. Polygyny, what do you mean by that? One particular character is controlled by multiple genes. That is what you mean by polygyny. So your skin color is controlled by multiple genes. Okay, and pleiotropy means one particular gene is controlling multiple characters. So in phenylketonuria, okay, phenylalanine is not getting converted to tyrosine. That is one problem. But there is one more problem. Melanin is not being produced, which is why the people who have this particular disease, they show hyperpigmentation, less amount of pigmentation. You get it? So pleiotropy, polygyny, everything is important, and whatever I told you as until now, all these topics are really important. Now this is a 13-mark chapter, which is why I am saying that I cannot say that 5 topics are going to come and that's all. I cannot do like that for these chapters. These are really important. If I miss one particular information, you are losing marks over there. Okay, you get it? Now one extra thing that I want to tell you is this. So over here, under Mendelian disorders, if you have the same book, I don't know, 73 page number, if you see these 2 pedigree charts, okay, just go through it once. Under that they have given the example of myotonic dystrophy and things like that. Just go through this is important. Okay, this particular image is also important. They will ask you what type of disease is this particular pedigree chart showing. So just for you to understand, if disease is found in every single generation, if you see diseased organisms, that means it is dominant. Okay, autosomal dominant disease. Okay, and it is not like the sex is not specified over here, and any sexes are having this disease and it is found in every generation, then it is autosomal dominant disease. If it is not found in the parents but found in the next generation, just like this, okay, just like this, if it is not found in the parents, oh, where is it? Yeah, in the parents but is found over here. Okay, so that will be autosomal recessive disorder, because this is recessive case. Only under recessive condition they will be showing it. Okay, so probably over here they had one dominant allele, which is why the disease is not shown over here. You get it? So I am taking this very seriously, this particular thing, which is why I brought NCERT and everything. I hope you are also following me really seriously and listening to me. These are the important topics, my dear children. Now let's get into the next chapter. Okay, next chapter again is of 13 marks, that is molecular basis of inheritance. Over here, DNA double helix model, this is a direct 5-marker question that can come. Okay, you have to tell about what is there. For example, one very important question, what is the difference between RNA and DNA in the term of its structure? Okay, major structure, nucleotide basically. So, DNA has thymidine or thymidine, thymine, okay, I will write thymine. DNA has thymine, whereas when you talk about RNA, my dear children, what does it have? It has uracil. So that is a question that comes straight, direct question, easy question. Okay, apart from it, you would be asked to explain the whole helix structure of what DNA. So how it is made, the sugar and the nitrogenous bases that is there, phosphorus group that attaches over there, the hydrogen bonds between the nitrogenous bases. There are 2 hydrogen bonds between adenine and thymine. There are 3 hydrogen bonds between guanine and cytosine. So that is all important. Okay, now haploid and diploid DNA content in humans, again really important. What is the haploid content, my dear children? It is 3.3 into 10 to the power 9. Okay, this is the haploid content. What is the diploid content? Diploid content is 6.6 into 10 to the power 9. So you should be knowing, they can give you in a gamete, human gamete, how much amount of DNA content is there. So it's a gamete. Gamete means half the number of chromosome. They will always have half the number of chromosome. That is haploid content. So your answer should be this. Okay, now structure of nucleosome really important, you have to draw it and show. Euchromatin and heterochromatin difference is being asked, so you should be knowing about both of them as well. Okay, which one gets stained better, which one gets stained better, which one is not getting stained better, where is transcription happening better, where is transcription not happening properly. So all these differences you should be knowing. Now 3 experiments in search of DNA. First one, okay, the first one was done by Griffith. Okay, second one, what was his name? Okay, just a second. Okay, I forgot. How can I forget? Okay, anyways, I'll tell. MacLeod and McCarthy. Okay, Avery, MacLeod, McCarthy. So those were the second people who did the experiment, and then you had what, Hershey and Chase. So these 3 experiments in the search of DNA is equally important. Give equal weightage to all of that and study it. Okay, features of genetic material, again this I can tell you where it is given in your NCERT because you might not be aware of it. So features of genetic material, just give me a second my dear children, even though I told you in wait, features of genetic material is given over here, 87 page number. Okay, if you have Hershey and Chase over here, you would be having 4 numbers over here. Okay, these are the features of genetic material that you should be knowing. It's on page number 87. So if you have your NCERT with you, just open it. Okay, you can pause the video and can open it and put a star mark over there. Now why DNA is better genetic material than RNA? That is also one thing. So because DNA is what? DNA is what? More stable, my dear children, whereas RNA is not. That is also given below this. Okay, below these 4 points that are given over here, if you are not able to see the 4 points, this looks somewhat like this. Okay, the zoomed in version would not be letting me focus things, but yeah, just below it you will be having, in this particular paragraph that you see over here, you will be having why DNA is better. So just go through it, because in the case of RNA you have 2 OH group. Okay, you have on the second position you have an OH group attached, which makes it ribose sugar. It is not deoxyribose, it is ribose sugar. Now this on the second position, the OH group makes it less stable. It is making RNA molecule reactive. Okay, it is making the RNA molecule reactive and things like that. So you should be knowing about that. Okay, all these paragraphs are really important, given on page number 87. Now we have the semi-conservative replication method and the Michelson and Stahl experiment which is given. So semi-conservative replication method, who gave? It was Watson and Crick only. And Michelson and Stahl, basically they did a experimental proof to it. So you have to go through the entire thing. You might be asked to explain it. Okay, so that is there. Now going ahead, what do we have? Structure of transcription unit. We have the structure of transcription unit, my dear children, which is given on page number 92. It looks like this. It looks, ayo, my positioning is totally wrong. Okay, if you can see, this is the thing. Okay, the structure of the schematic structure of transcription unit, that is really important. You should be knowing where does the rho factor and the sigma factor come and attach, what is rho factor, what is sigma factor, everything, every single thing. Now splicing and the types of RNA also which is there. Again, did I miss something out? Just a second, okay? Okay. Now, yes. Splicing and the type of RNA, what exactly is splicing? This is again a 1 marker question that comes. Okay, so you should be knowing it's a removal of what, introns, and joining of exons, which is known as splicing. And the types of RNA which is there, you have different types of RNA in the case of eukaryotes. So some RNA is there for RNA polymerase 1. Okay, not RNA, it is RNA. Just a second, I'll make a quick change over here. So it is RNA polymerase, the types of RNA polymerase. So you should be knowing that RNA polymerase 1 is responsible for transcribing rRNA, polymerase 3 is responsible for transcribing tRNA, snRNA, things like that. RNA polymerase 2 is the one that transcribes the DNA into hnRNA and then later into mRNA. So all this little piece of information is important. Again, you can find it on page number 94 above this particular image that you see over here. Okay. Why is it going? The camera is right over here. Okay, fine, cool. Anyways, so that is really important. Okay, now features of genetic code. Now that is a whole different thing. You have it readily in your book. Just go through it. Very important. Can give you lots of marks every time it is asked. Okay, so you can expect a question coming off from there. Translation, again, each and every part of it is important. I'll tell you what is more important over here. That is amino acylation. Okay, what do you mean by amino acylation? That is really important. Okay, what is the first step of translation? That is important. What is the structure of tRNA? Structure of tRNA, where does the amino acid come and attach? It attaches to the 3' end of the tRNA. The amino acid comes and attaches on the 3' end. All this little piece of information is really important. Okay. Now, regulation of gene expression in eukaryotes, my dear children. Regulation of gene expression in eukaryotes, how will you study this? Now, this is given on 99 page number. If we see the regulation part, okay, if you see the regulation part, there are 4 points again given over here. So, you should be knowing all these 4 points. Okay, page number 99. It is really important. That is, it regulated at transcription level, processing level, transport of mRNA from the nucleus to the cytoplasm, and also translational level. Cool. Now, you have lac operon entirely, it is very important. Human Genome Project, again you can be asked about the goals of Human Genome Project and also the findings, the salient features of Human Genome Project, both. Okay, Human Genome Project means you can be asked about goals or the salient features. Cool. Now, DNA fingerprinting or the use of VNTR is equally important, again something that usually comes in. So, all this information from this particular chapter is important. Please go through lac operon really well. Always there is a question, I do not want you to miss it. Okay. Even transcription, you need to learn to draw it. If you have not seen the most important diagram video, go watch it. There I have mentioned about the most important diagrams of biology. So, there you will get a lot more information about the diagrams also that could come. Okay. Here I have not included diagrams. So, yes, that is there. Apart from it, okay, DNA replication. Which one is lagging strand? Lagging strand. Which one is leading strand? In which direction does DNA polymerase move? It moves from 3' to 5' direction of the DNA. If DNA is in this direction, then the DNA polymerase will be moving from 5' to 3' direction like this, opposite to this. So, this is the DNA strand that they want to make a copy of. So, this DNA polymerase comes and makes a copy from 5' to 3' direction, but gives reference to this, okay, gives reference to this, but it itself goes from 5' to 3' direction. So, please make sure you are understanding that. Okay. Okazaki fragments, all this piece of information is really all the enzymes which are there, DNA ligase, DNA helicase. Helicase is for breaking and opening it, okay, and forms a replication fork, and DNA ligase is for joining the joining the later segments of DNA which are there. All these things are really important. Okay, cool, works. Chalo. So, yes, there you go. Now, human health and diseases, for this I do not need NCERT at all. Okay, I can just say as it is. So, bacterial, viral and protozoan disease, all the diseases, okay, that is your typhoid, your pneumonia, that is coming under bacterial, and also diphtheria is coming under bacterial disease, dysentery, normal dysentery, okay, not amoebic dysentery, normal dysentery is coming under bacterial. So, all these things you should be knowing. The name of the disease, name of causative agent, symptoms and how it is spread. These 3 information about all the diseases given initially in your NCERT is really important. In viral comes one, that is what, your common cold. Okay. Protozoan disease, you have malaria and amoebic dysentery. Okay, and then you have fungal, I do not know why I did not mention that, you have fungal and helminthic diseases as well, fungal and helminthic disease. So, I am writing helminthic diseases over here. Helminthic diseases, you have what, filariasis as well as ascariasis. In the case of fungal, you have ringworm disease. So, again all these diseases you should be knowing. Okay. Now, under innate immunity, just go through innate immunity, each and every point of innate immunity, cellular defense, that defense, this defense, physiological, psychological, things like that. There are so many things, right. Now, under that, interferons is much more important, very much important. So, interferons are being released by viral infected cells, my dear children. So, you should be knowing about that. Now, acquired immunity, in that acquired immunity you should be knowing about humoral and cell mediated immunity. The structure of antibody is very important. I told you in the most important diagram video, but I am telling you again, very, very, very important. Every time it comes. Okay. Now that is there. In active immunity, okay, and one more thing, in graft rejection, which one plays a major role? In graft rejection, which one plays a major role? It is cell mediated immunity that plays a major role. Understand that. Active immunity and passive immunity, examples can be asked. A man is being bit by a snake, and then the doctor gives anti-venom already been prepared. What type of immunization is this? This is passive immunization, because you are giving ready-made antibodies. Okay. Active immunity, where can you see it? When you get a disease, what immunity you develop, that is known as active immunity, because your body is actively participating in producing what? Antibodies. In vaccination, this is the theory. Why do you give vaccine? Vaccine means live attenuated, a form of pathogens. When they are inserted, okay, they are anyway, they are not in a state that they could cause infection. Okay, they are inserted in our body, and our body will react to it, such that when actually something comes on, we can react to it. You get it? So that is active immunities. Example, vaccination is given. Now there is antibodies, two different antibodies that is being mentioned over here, IgA and IgG. So passive IgE and IgG. In passive immunization, I told you already give ready-made antibodies. Where can you see it in human body happening naturally? In the case of mother and child, the mother gives directly the baby immunoglobulin A via milk, colostrum. So colostrum is very rich in that. So this is an example of what? Passive immunity. Similarly, IgG is given through placenta to the baby. Again, this is the first antibody that goes through. We have 5 antibodies, GAMED. Okay, A and G are being passed from mother to the child initially itself. Now in allergy, what do we have? You have mast cells, my dear children, releasing what? Histamine. So you should be knowing that releases histamine and serotonin, and IgE is the one who plays a major role in allergy. Lymphoid organs, the primary and the secondary lymphoid organs, all of them are equally important. You should be knowing it, each and every one among them. Okay, spleen, lymph node, this, that, everything. HIV and cancer, again really important. How is HIV spread? Okay, what is that particular test that can be acquired for testing HIV? That is ELISA, my dear children. What has the government done about it? What is the factory of HIV? It is macrophages. Which cells are mostly affected in HIV? T lymphocytes. Which T lymphocytes? T helper lymphocytes. So all this piece of information is important. Cancer, again a very important topic from this particular chapter. Okay, the causes of cancer, how cancer cells are different from normal cells, what is the treatment method available for cancer, how can we find that someone has diagnostic method available for cancer. Each and everything under cancer is important. Okay, so go through it really well. Now name of the source, drug and its effect. In drug, opioid, cannabinoids, coca alkaloids, barbiturate, amphetamines, everything, each and everything is important. You should be knowing its source, that is from which plant you are taking. Okay, for example, opioids you are taking from opium poppy, or what do you call it? Somehow I forget things. Oh, I was going to say Pisum sativa. God forbid me. Okay, wait. Papaver somnifera, yeah, papaver somniferum, that is from where you get opium poppy. Okay, so that is important. Cannabis sativa is important. Okay, and erythroxylum coca is important. So all these plants are really important. Also opioids, it affects your central nervous system, CNS, and also your gastrointestinal tract, whereas cannabinoids, it affects your cardiovascular system. Okay, when you talk about coca alkaloids, it affects your CNS again. So all these effects are also really important. Nicotine is going to increase the blood pressure and things like that. You should be knowing about that as well. Drug and alcohol abuse, you can just go have a read of it, but these are the important topics that you have. Cool. And Brahmastra series, go watch it as well. Now yeah, so we are completing human health and diseases as well, which carries around 11 marks. Yeah, and now okay, we have sexual reproduction in flowering plants, again something that carries 11 marks. Okay, so in that, structure of microsporangium and the pollen grain, equally important. You should be knowing about that. In microsporangium, the different layers that you are having. Okay, the middle layer, the tapetum, and epidermis, endothecium, each and every part of it. Okay, in pollen grains, the intine, exine. What is exine? Having sporopollen, very much important. What is that resistant substance? Sporopollen, which can thrive through anything. So you should be knowing about that. What is the structure of the nucleus inside the pollen grain, or the cells inside the pollen grain? So you have vegetative and generative cells, so you should be knowing about the structure of the nucleus, and also how many percentage of them release the pollen grains in 3 cell stage, and basically how many of them release them in 2 cell stage. Each of that information you should be knowing. Okay, what is microsporogenesis? Explain. So you should be knowing how to explain microsporogenesis. Structure of megasporangium given in your NCERT. You should be knowing about the structure of megasporangium, my dear children, with each and everything, integuments being mentioned, funicle being mentioned, hilum being mentioned, placenta being mentioned, each and every single part. Micropyle, chalazolant, each thing. Okay, embryo sac, again very important. The diagram is really important. The cells which are there are really important. You should be knowing that they are haploid. Okay, so megasporogenesis is also equally important. Now pollination, you have autogamy, geitonogamy and xenogamy. In that, autogamy, my dear children, is really important. Given the example of clistogamous flowers, oxalis, viola, commelina, all of them. Okay, so you have clistogamous flowers and you have chasmogamous flowers together in one particular plant, that is the viola, oxalis and commelina that I was talking about. So you should be knowing about that. Geitonogamy and xenogamy, which one gives let's say distinct progenies, the progeny that has lots amount of variation, it is what? Xenogamy, my dear children. Rest of them, both of them are self-pollination itself. Okay, now pollinating agents, you have wind pollinated, water pollinated, insect pollinated thing. Mostly, okay, mostly I am saying wind and insect has been asked, but this time anything else can be asked, that is water can also be asked. So you should be very well prepared to it. Okay, so they can ask you about the features of the flowers which undergo wind pollination, or the features of the flowers which undergo insect pollination and things like that. They have asked like that. So this time it can be for water as well, so go through it really well. Outbreeding devices, that is the one that helps the plant understand that you should not be inbreeding, you should be doing outbreeding. Okay, so these devices are really important every time it is asked. Artificial hybridization, my dear children, where emasculation, bagging and everything is there, just go have a quick read of it. It will be good. It's an easy thing, you will be able to understand it easily as well. Okay, now double fertilization, all the events in there, syngamy, let's say triple fusion, everything. Okay, what do you get after all this double fertilization? You get a zygote, and also you get what? Primary endosperm cell. Okay, and this will keep on dividing, forming endosperm and forming embryo and things like that. So go through it really well, and then okay, and then you have structure of monocot and dicot embryo, my dear children. The structure of monocot and dicot embryo, and also before that what all stages are there, globular, heart shaped and things like that, pro-embryo, everything, all the stages which is given in your NCERT, you should be knowing about that. So I am just writing for you to remind yourself. Pro-embryo, okay, globular, heart shaped, everything you should be knowing. Okay, the stages through which the embryo passes. Okay, and the structure of monocot and dicot you would be asked to draw also. You should be knowing about all single thing, coleorrhea, coleoptile, and then scutellum in the case of monocots. Okay, in the case of dicot, the 2 cotyledons which are there, and all these different parts. It's really understandable. Okay, and then you should be knowing non-albuminous and albuminous seeds, my dear children. Okay, what is difference between pericarp and perisperm? How did I miss it? So pericarp is the outer covering of the ovary, and then you have and then perisperm, the remnant nucellus which is present, which provides nourishment. All these examples you should be knowing. Okay, examples and the definition for all this. Parthenocarpy, where do you find parthenocarpy and things like that. Apomixis, again something very important, asked most of the times. Why apomictic seeds are better? How does it help the farmers? Given in the last part of your NCERT, just go through it. Okay, fine. In the perisperm, they will ask you about the examples. So it is beet. It is beet as an example. So just go through it. All these small black pepper and beet are the examples for this. You should be knowing its examples as well. Okay, cool. Rest under images also there are, as I said, there are important things. I have explained in the most important diagrams. Just please go watch it, because if I make everything all together over here, it will be too much for you to understand, and you might feel that, oh my god, I am not reached anywhere. So I don't want you to do that. Just go through it clearly. Yeah. Now in human reproduction, my dear children, male reproductive system and spermatogenesis is really important. You might be asked to draw the schematic representation of spermatogenesis, where the cells are being divided. Okay, primary spermatocyte, secondary to spermatids, then to zoa, and then spermiation happening, all these things. Okay. Now female reproductive system, again the picture can be asked. Oogenesis, you might be asked the structure and like schematic representation of that as well. And also mainly the questions comes around secondary oocyte, my dear children. Secondary oocyte releases what? Zona pellucida. So that is very much important, zona pellucida. So when it is released, it is in which state? It is in this state where secondary oocyte is there. It has not completed meiosis. It is stuck in meiosis 2. It is stuck in meiosis 2. Please remember that. Okay. Menstrual cycle and the difference between menstrual cycle as well as estrous cycle, that has come. Okay. Fast and slow walk of polyspermy after the fertilization happens. Okay, sorry, before the fertilization happens, you have so many sperms coming together. So you have to block certain sperms, right? Only one can fertilize. So that is given in your NCERT. Just go through it in under fertilization part. That is important. Okay. Now cleavage and implantation, in which stage does implantation happen? In blastocyst stage. In blastocyst stage, implantation happens, my dear children. So you should be knowing about that. Placenta's function and its hormones. There are 4 hormones released by placenta. So you should be knowing about that. The functions of placenta, you should be knowing about that as well. Okay, exchange of gases, removal of waste materials and stuff like that. Hormones, you know, human chorionic gonadotrophin, human placental lactogen, you have estrogen and progesterone. Okay. Apart from that, there is something that is known as relaxin being produced by ovary. So that can be a question, which is that thing that is produced by ovary during later phase of pregnancy. It is relaxin, my dear children. Okay. So there are other hormones also which are increased in multiple folds during pregnancy. Estrogen, progesterone, prolactin and things like that. Thyroid hormones. So you should be naming them also. Okay. Now stages of development of fetus, what happens in the 5th month, what happens in the 3rd month and things like that. All those periods are really important. Fetal ejection reflex plus parturition is important. Let this baby moving out. So what is fetal ejection reflex? How we can say that both the nervous system and the endocrine system plays a major role over here. So you have to say the signal comes from the baby and the placenta, and then in the body the estrogen receptor, oxytocin receptors are increased in the myometrium. Oxytocin is being released, oxytocin comes and attaches over here, causes contraction, and again a signal goes, more oxytocin is being released, and the whole cycle goes on. So explain it really well, and if you are not able to explain it properly, I am there for you. Go watch my Brahmastra series. Why I am telling you is that we put a lot of efforts into it for finding questions which have come repeatedly, okay, which is why I am standing here, okay, without probably with lots of recollection of things, because we did it so many times. You get it? So this is why I am telling you, trust me on this and go through all these things. Have your NCERT with you. Go watch the Brahmastra series at least once before your exams. Okay, so you should be definitely giving it a watch. It will take only 1 or 2 hours probably from your day, or probably 1 hour in some chapters. So utilize that time, put in 2x speed, just watch it. Okay. So those are the important topics from this particular chapter. Now what? Microbes in human welfare, there you have sea-based treatment, my dear children. Okay, so you should be knowing about the primary treatment, secondary treatment, what happens in that, what is BOD, biochemical oxygen demands, what is flux. Okay, all these things are really important. Biogas production and plants, so you should be knowing about the biogas plant also, who plays a major role over here, methanogens, my dear children. Here also in secondary treatment, who plays major role? Aerobic bacteria first, and then comes anaerobic bacteria also. Okay, there also biogas is being produced. About the primary effluent, secondary effluent, all those things, activated sludge, all this little piece of information is really important from here. Now biocontrol agents and biofertilizers, all the little tiniest examples of biocontrol agents, my dear children, whether it be Trichoderma polysporum or ladybird or what do you call it? Dragonfly, all the things that are given in your NCERT as biocontrol agents, you should be remembering it. Similarly goes for biofertilizers as well, Nostoc, Annabena, all the cyanobacteria that you have, all the things that are given in your NCERT which acts as biofertilizers, look for the examples for them. So I am just writing examples, just know the examples of each. Now statin is being released by what? Monascus purpureus. Cyclosporin A is what? You should be knowing statin is the one that stabilizes blood cholesterol level or makes it lower. Okay, cyclosporin A is an immunosuppressive agent released by Trichoderma polysporum. Streptokinase is released by Streptococcus. What is streptokinase? It's a clot buster. So each functions each of them you should be knowing their functions and also their source. So I am writing over here functions and source. Okay, now acetic acid is released by Aspergillus niger, citric acid by Acetobacter aceti, okay, butyric acid by Clostridium butylicum, lactic acid by Lactobacillus. Again, you should be knowing the source over here. Roquefort cheese, how it is ripened with the help of fungus. With the help of fungus, how it is ripened? With the help of fungus. Curdling of milk, how it is possible? With the help of lactic acid bacteria, my dear children. Okay, penicillin is an antibiotic released from Penicillium notatum. What is this? This is again a fungus. Okay, this is a fungus. What do we have? One more fungus, Saccharomyces cerevisiae, which is what? Just yeast, my dear children. Whatever function it has in the case of fermentation, in the case of Swiss cheese, okay, in the case of forming big holes in Swiss cheese by release of carbon dioxide, in the case of brewery, everything you should be remembering. Okay, so all these examples, if you remember, you think half of your work is already done. Cool. So how will you be able to remember, my dear children? Only by practicing. Otherwise you won't be able to remember. Otherwise it will be cool. Now in evolution we have S.L. Miller's experiment who gave the proper construction of the chemical evolution. So you might be asked to draw the diagram as well. So go through it really well. Then you have convergent and divergent evolution. They can ask you by giving examples. They will ask you, okay, Bougainvillea has thorn and cucurbita also has thorn. Sorry, cucurbita has tendrils and things like that. Okay, what type of evolution do they show? Convergent or divergent? So it is what? Basically divergent. Both of them had a common ancestor. From there they got diverged because of different habitats. So you should be knowing about each examples. With examples they can ask you. Now industrial melanism is an example that is very much important. You should be knowing about that. Okay, about the name of the moth that was being used, Bistellaria and things like that. Okay, now adaptive radiation, Darwin's finches, Australian marsupials and placental mammals. 3 of them are given in your NCERT and 3 of them are equally important. Just go through it once. Okay, Hardy-Weinberg principles and the factors affecting it. Hardy-Weinberg's principle, you should be knowing about this formula that is there. Okay, you should be knowing about that, the factors affecting it, genetic drift, this, that, okay, gene migration, all these features are really important, which is there. So you have to go through it. You would be asked to explain this really well. So Hardy-Weinberg principle is important, and in that category only comes this particular, there is a picture where they are asking you stabilizing, directional and disruptive things. Okay, if you want I can tell you on which page number it is so that you don't have much amount of confusion there. Just give me a second, okay, because I obviously do not remember the page number, but I can tell you. Evolution, okay. Okay, it is on page number 120. Okay, where they are telling operation of natural selection on different traits. So this is the picture that you have. Just go through it really well once. Okay, if you can see it. Yeah, so just go through it. Stabilizing, directional and disruptive. You might be given the picture and you might be asked how to frame it. So frame questions I have already put in Brahmastra. Just go and watch it. You'll get an idea of it. So these are the things from this particular chapter. Apart from that, role of lowfins and shrews in evolution. So lowfin is a link, that a kind of link between what? Your Pisces, fishes as well as amphibians. Shrews is a link between reptiles and mammals. Okay, now evolution of man, Homo erectus and things like that, all the different Ramapithecus and then Australopithecus, Habilis, okay, all the different things you should be remembering them order wise, year wise as well. Okay, it's easy, trust me, it's easy, just have to go through it. I'm telling you, 12th biology is really easy. If you have passed 11th biology, then my dear little children, put a big tap on your back. That is much more difficult. Okay, so 12th biology is kind of easy. Just trust me, you can do it. Okay, chalo, next. Now we have biotechnology principles and processes, my dear children. So over here we have restriction endonuclease and exonuclease. You would be asked to differentiate between that. Okay, Hind2, Eco R1 are very important. Hind2, how many base pairs does it have in its recognition site? It is 6 base pairs. What about Eco R1? Same thing, but you will be asked the base pairs also. It is GAATTC. That is the code. Okay, that is the recognition site. 5' to 3', that is the way. Okay, now gel electrophoresis, each and everything about it. Also, what is stain is being used? Ethidium bromide under what? UV radiation, my dear children. It cannot be viewed without UV radiation. Features of a vector, that is origin of replication. What is the use of origin of replication, my dear children? Because it controls obviously replication and also the copy number. That is important. Selectable markers, the cloning sites which are there, that is also really important. PBR 322, the diagram of it is important. Each and every function of it is really important. Insertional inactivation about beta galactosidase, that example that is given, really important. Okay, over here also ampicillin resistance, tetracycline resistance, all this acts as a marker. Okay, now how to make cells competent? By giving heat shock treatment. Biolistic method is there for plants, microinjection is there for animals, disarmed retrovirus is there for animals again. So you should be knowing who is going to act like vectors and things like that. So if it is vectors and if it is for animals, then it has to be disarmed retroviruses, my dear children. Okay, if it is plant, then it is agrobacterium tumefaciens which has TI plasmid. What is the use of TI plasmid? It is a tumor inducing plasmid, but we have removed the tumor inducing part and we have inserted a gene of interest. Okay, now isolation of DNA, the enzymes which are there. For example, you have chitinase, you have what? Beta, cellulase. Okay, all this lysozyme. So all these things are there for breaking down the cell wall. Then for clearing the RNA out, you have ribonucleases. For clearing the proteins out, you have proteases, and then finally you get the DNA. How do you separate it? By adding chilled ethanol. So all these enzymes and everything is really important over here. Now apart from that, why did I add this particular thing? I don't know. Now polymerase chain reaction, again the order denaturation, then annealing, then extension. Important. Okay, what polymerase is being used? It is Taq polymerase which is being used, and what temperature and everything you should be knowing. Bioreactor, you will be asked to make the diagram of it. Downstream processing, you will be asked just to write something about it. Okay, what is downstream processing? The last process that you have, purification and things like that, processing of the content that you are getting from the whole process of recombinant DNA technology works. So you should be knowing about all this thing from the chapter biotechnology principles and processes. Cool. Now going ahead with the next one, biotechnology and its application, which is a 6-marker chapter, my dear children. So over here, features of GMP. What do you mean by GMP? Genetically modified plants. That is also given initially. Okay, in that they have mentioned about golden rice and things like that. So just go through it. BT cotton and the cry gene. So you should be knowing which cry gene is responsible for killing cotton bollworms, which one is responsible for corn borers. There are two ones, corn borers and cotton bollworms. RNA interference method, it is the method for eukaryotes, not prokaryotes. Okay, it is eukaryotes. In that, Meloidigan incognitia is being eradicated with the help of that, not eradicated completely, but yes. So RNA interference method, just go and read it. It is affecting the tobacco plants. So how it is affecting, you should be knowing it. Okay, so in RNA interference, double-stranded RNA is being used. Always remember that. Okay, now insulin is being made in our body, but if we do not have enough insulin produced, basically you make it produce outside in the laboratory condition and things like that. Okay, with the help of E. coli and things. So here Eli Lilly, a company, pharmaceutical company, had a major role to play. You should be knowing about what role did they play. So there is A peptide, B peptide and C peptide. You don't want C peptide and things like that. So there was huge amount of inventions and things being done by Eli Lilly company. So just go through it. Okay, now gene therapy, ADA deficiency. What was gene therapy all about? What do? You take lymphocytes, basically you insert that particular gene and then you put that in lymphocyte back in the body, and then after that some point the lymphocytes die, so again you need transfusions of this. So this is all about gene therapy, so go through it as well. Okay, now PCR as a tool for finding trace amounts of pathogens. So if you do not know, let me tell you, if you want trace amount of pathogens, if you just have trace amount of pathogens in the body and you still want to find the disease, Okay, with the help of PCR, what you can do, you can amplify it, and then you can find, oh, you are actually infected, you have this particular disease. So PCR is one such tool, remember it, very, very important. Okay, biological products of transgenic organism, for example, alpha-1 antitrypsin, you have alpha-lactalbumin, all these things given in your NCERT is really important. Just go through it. Under this topic you will be finding. Okay, transgenic organisms, if you see at the end of the chapter, under that you have biological products from it. So in that category you will be finding it. Now GAAC and also biopiracy, about the basmati rice and things like that. What is exactly biopiracy? Just go through from your NCERT. Okay, biopiracy is basically when you are using someone else's product without their pure knowledge, and basically you are not asking any permission, you are just doing it and you are putting patency on it. Okay, that is what you mean by biopiracy. You are explaining that this is mine, but this is not actually yours. You get it? Okay, wait. No. Now what do we have? Reproductive health, my dear children. In that we have first thing, amniocentesis. Amniocentesis is used for what? Female feticide. That was the problem that was there. Okay, what is amniocentesis? You should be knowing about that. Infant natality and infant and maternal mortality rate. How does it affect the population growth? You should be knowing about that. Okay, for example, if natality is more, that is birth rate is more, and death rate is less, that means population growth will be happening. If mortality rate is more, that means population is not growing, it is going down. Okay, now contraceptive methods, you know there are so many methods. Under that I will recommend you to specific, for example, you went through everything once, but I want you to go through IUDs and oral pills and surgical method twice in that also. Okay, now MTP is medical termination of pregnancy. Just go through it, my dear children, why it is not recommended or when is it safe and things like that you should be knowing. Now incurable STIs, under sexually transmitted diseases there are certain incurable ones. For example, you have genital herpes, okay, and you have hepatitis, and then what was it? HIV. So this is all incurable, all of them starts with H, so you can remember like that. Now steps for preventing STIs, that is also given in 3 points, very much important. ART, under assisted reproductive technologies, my dear children, you have ZIFT, GIFT and ICSI and artificial insemination, lots and lots of things, but mainly if you are asked about ZIFT, okay, so you should be knowing where are you putting the embryo into. If it is in 8 cell stage, you can put it in the fallopian tube. If it is more than 8, then probably you have to put it in the uterus. Okay, so know about the small information details which are very, very important. Cool, okay, chalo, let's go ahead. Okay, now what do we have? Organisms and population, my dear children. In that, pyramids, you should be knowing about expanding, stable and declining population pyramids. You will be asked, you will be given the diagram and you might have to find one which is expanding, which is stable and declining. You have to see the pre-reproductive, post-reproductive population and things like that. Okay, now factors affecting the population growth, that is natality, mortality, immigration, emigration, all these factors you can be asked to write about all of them. Exponential and logistic growth, my dear children, here you get J-shaped curve, here you get S-shaped curve, just go through it. Now you have different type of interactions between organism, predatorship, competition, parasitism, commensalism, ammensilism, all of it you have to go through obviously, but in predator please have some examples in your head. Okay, such as Pisaster sea starfishes, or basically you can you have to also you have to anyway definitely remember the defense mechanism against the predator. For example, monarch butterfly is distasteful and things like that. Acacia plant have thorns. So these are plants which have defensive mechanism. You are asked about plants and animals separately. Plants and animals, what are the defensive mechanism against what predators, that is being usually asked. So you should be knowing about that. Okay, in competition you should be knowing about the Balanas example and the competitive Gauss's competitive exclusion principle, my dear children, very, very, very important. Okay, and then parasitism, you have ectoparasites, you have endoparasites, you also have brood parasitism, and that is very much important. And I have put sexual disease also over here because the question comes like this: explain brood parasitism and sexual deceit. Even though sexual deceit does not come under parasitism, but still I am telling you over here itself. So the example is about the orchid, that is Ophrys, which let's say portrays it as the female bee, which is why the other bee comes and tries to basically fertilize or basically undergo the process of sexual reproduction, but apparently it is just a orchid that is there. So that is sexual deceit. It is being, let's say, cheated on, like that. So example is really important, definition is really important, just go through it. Okay, so here also whenever I am saying just go through the example, go through the examples, my dear children, very important. I do not know how to say how much important, but yes. Now in commensalism you have egret and cattle, you have epiphyte and mango tree, and you have sea anemone and clownfish. These are 3 examples of commensalism. In mutualism you have lichens, you have mycorrhiza. Each and every part of it is important. When I say about organism and population, it's about examples. It's mainly about I wrote enzymes. You see, my brain has gone, oh my god. So that is how my brain is processing as of now. So I understand how your brain might be processing, because you have to study this as well. I have already studied it. So, but children, I am telling you, listen, this happens when you are not calm. So I am not calm. Why? Because it has already been too long. That is why I am not calm. Okay, I don't want to go any more longer, which is why I wrote enzymes. So in your case also, please be calm and study, otherwise nothing will go into your brain. Okay, so these are the important things from organisms and population. Cool. Now, what do we have? Okay, we have ecosystem, that is the second last chapter that we have. Okay, in that components of ecosystem, my dear children, you will be asked to define all of them: productivity, decomposition, energy flow and nutrient cycling. Or you can get an MCQ question which says find one which was not the component of ecosystem. So they can give you something else instead of one. They can shift one and they can put something else. So you have to find that. Now productivity, GPP and NPP you should know, gross primary productivity and net primary productivity. Decomposition, very much important. Okay. You can expect a 3-marker question, not a 4-marker question, because 4-marker question is not there. Okay, so basically I was thinking, yeah, so it is a 3-marker question that you can expect from decomposition. You can be asked to explain the whole thing, or in parts also they can ask you in 2-marker or probably 1-marker. Okay, so steps and the favorable conditions which are needed. What are the favorable condition? Moist environment, oxygen is required, temperature should be high, all these are the favorable conditions. Okay, now in steps you should be talking about catabolism, leaching, mineralization, humidification, all these things. Okay, grazing food chain is most prominent in aquatic ecosystem. I have just written over here so that you don't forget. And photosynthetically, okay, grazing food chain in aquatic ecosystem is really important. And whereas when you talk about terrestrial ones, in terrestrial, what is more important, my dear children? In terrestrial, major amount of energy flow is through detritus food chain. Detritus food chain, please remember it, you can get the question. Now photosynthetic active radiation and standing crop, just go through it, it is really important. Just go through what it's trying to say. Okay, definition, only 2 to 10% of the total solar energy which is there, photosynthetic active radiation, is being taken up by the plants. So you should be knowing about that. What is standing crop? It is a total biomass available at a particular level. Yeah, so yes, that. Now what? Pyramid of number, biomass and energy. You should be knowing about it. Again, this is star portion. Pyramid of number, the biomass as an inverted, which one has inverted biomass? In aquatic ecosystem you have inverted biomass. And energy's pyramid is always upright, it was always straight. So that is also really important. Cool. Now last but not the least, you have biodiversity and conservation, my dear children. Over here this image is really important, this image is also important. So tropic has more diversity than temperate regions. You would be asked why, you have to explain it. Okay, species area relation formulas, that is this. So just go through this, important. Evil quartet, as in the 4 reasons why there is biodiversity loss, all 4 of them are really important, just go through it. In that, one is overexploitation, stellar sea cows example is given. This example is really important, this was not there, this is not there as of now because of overexploitation, you should be knowing about it. Alien species invasion, this is also one such part of evil quartet. Now Nile perch was introduced when, so which is why the cyclid fishes which were there, they got extinct. Okay, Parthenium, lantana and water hyacinth are another plant things which were introduced and which made the other plants devoid of lot of things. Okay, so these are alien species invasion example. Now broadly, narrowly and ethical arguments for conserving biodiversity. In that, narrowly utilitarian is the most important one. In situ conservation, sacred groove is really important. Ex situ conservation, everything is important. Just go through it once. You will be asked what is not a type of in situ conservation, okay, or ex situ conservation. So seed bank and everything will be given as example. You need to find out. So if you are not aware about in situ conservation, you will not be able to do that as well. Works? So my dear little children, are you ready? I know it's too late to ask, but yes, are you actually ready? I hope I explained each and everything to you in much detail so that you don't miss even a single mark, even half mark or 0.25 marks. So I hope you're ready. If you're ready, do let me know in the comment section. I don't want to waste your time, so I'm just going ahead and saying tata bye bye for the day. Definitely do not forget to subscribe to our channel and also put down comments if you like this video, and do let me know if you need any help. I'll truly try to make this video really short next time if I'm doing any such videos. Yeah, so that's all. Bye bye, take care.